fiber is narrow. What happens? There is fibrosis instead of entire replacement. And if you look at the progenitor cell, it is sleeping all the way in the health, in the diabetic sector, the pink color. Whereas in the other one, it becomes yellow and becomes active and then comes to rest. So all these abnormalities do happen in a muscle fiber, and this might also contribute to the formation of diabetic myopathy. Coming to satellite cell impairment, this is what we have been just now discussing. The satellite cell or the MUSCs are the same, and they are the cells which are quiescent initially, gets activated, then differentiates into myocyte, myoblast, then myocyte and myotubes. It is really shocking to see that diabetes suppresses at all conversions. So regeneration or recovery becomes very, very difficult. And the good news is that physical activity or exercise is able to help all these things. So in that way, we should understand one more advantage of exercise. In a nutshell, talking about what all I've been talking so far is atrophy on one end and metabolic dysfunction on the other. When these two are again a vicious cycle happening, it is mediated through insulin resistance, poor glucose intake, as well as poor protein intake and protein formation, more damage to the protein, and mitochondrial damage. And this also is associated with lipids settling down along with the muscle fibers, and which produces again further inflammation. And this inflammation produces reactive oxygen species and also advanced glycated products. So all these things put together cause the continuation of this vicious cycle. Coming to sarcopenia, as I told you, muscle atrophy and poor muscle function is diagnosed as sarcopenia. And this can be worsened by intramuscular deposition of fat leading to poor muscle function. I'll tell you, fat which is entering into a muscle cell is beneficial for the cell. But the fat is not entering into it, but lying by the side of the muscle in the different layers. What you see it in the broiler chicken, this is what is seen in the broiler chicken, and they create insulin resistance. That is why you have the bulk. And here, this particular fat is always dangerous, and that is called sarcopenic obesity. Undernutrition and loss of body weight hastens a decline in muscle mass and strength. Sedentary behavior, age, due to illness, disability, or lifestyle, further increases the risk of muscle loss. Decreased muscle strength, reduced working speed, fatigue and impaired balance, and also increased risk of falls are the symptoms of sarcopenia. How do you diagnose sarcopenia? Very simple test I'll just mention. You just measure the calf circumference. If it is going to be less than 34 in men, less than 33 in women, probably they are sarcopenic. There is one more correlation with waist. So it's called waist-calf ratio. If it is going to be more than 2.4, it is a drawback, that is they are going in for sarcopenia. And one more chair test is also easier. Within 30 seconds, the person should sit, stand for at least 5 times. If he is not able to do that, that means he has got sarcopenia. So this can be diagnosed even with a single pie. Probably what you need is a knowledge. And what are the mechanisms potentially contributing to sarcopenia that may be amenable to modification with drugs? You look at various pathologies, and the good news again is we have drugs to combat all these things. So coming to the current treatment strategy, exercise acts on the muscle, releases a lot of myokines, and all these myokines shown in the green arrow are favorable, except myostatin, which reduces the muscle bulk. Thanks to the liver, which again mediated through exercise, helps by follistatin, and which is able to suppress myostatin. Thereby, with exercise, you get the best benefit. What about sleep? Better quality and quantity of sleep at the right time. It is not a 2 a.m. to 10 a.m. It should be the stipulated time. It's important for a muscle balance. Food, MUFA-rich and SAFA-rich should not be used for reducing insulin resistance. Adequate protein intake with 1.2 grams of protein per day, and should contain leucine, isoleucine, argine, mainly non-vegetarian food like meat should be a little less, fish, chicken, and then nuts, dark chocolate. All these things contain all these needed entities. Coming to pharmacological intervention, insulin, GLP-1, DPP-4 inhibitors improve muscle mass. Sulfonylureas, negativate. Rest of the things, not very sure, but this is along with exercise and euglycemia. These are the drugs that we routinely use, and they have several mechanisms of action. And vitamin K2 is again a drug which alleviates insulin resistance. ARB, ACI inhibit, we have always been using it, but the recent study on rats say that irbesartan is better than even exercise in reducing insulin resistance. Probably if that is so, I would just gulp irbesartan and lie down peacefully and do not do any exercise. What are the pipeline treatment options? Mitochondrial transplantation. You see on the left a young lady walking around, and you see another person with sarcopenia on the right with a defective or a dysfunctioning mitochondria. Change it, very simple, and then probably in days to come, the mitochondria restores the muscle bulk, muscle health, and then you you can be like this particular lady. So, well, little limited data at present. Hexaprotein or the hexosaminidase A is again a rat model which has been showed with increased glucose uptake. And lastly, myo-DN. This is a muscle-forming oligo-DNA. So you inject into that particular muscle, it binds to the target protein, nucleolin, in myoblast and improves differentiation of myoblast. What diabetes does is stop the progression of myoblast to muscle cell, whereas this improves it. So in that way, probably we could bypass the effects of drawbacks of diabetes. So dear friends, to summarize, skeletal muscle and diabetes have bidirectional relationship impacting each other. The role of insulin on skeletal muscle is vital, without which a lot of derangements happen. Inadequate or inefficient action of insulin on muscle in diabetes causes hyperglycemia and also muscle dysfunction. With good muscle mass, strength and function, achieve freedom from sarcopenia. Inflammation, oxidative stress, satellite cell impairment, mitochondrial dysfunction are some of the pathogenic mechanisms in muscle dysfunction. Simple muscle fiber alteration from type 1 to type 2 itself causes poor muscle regeneration. And not only damage to muscle, but impaired skeletal muscle regeneration happens in diabetes. Lifestyle modification with adequate exercise, quality sleep, sleep in the right time to the right duration, and healthy diet with more protein is useful apart from glycemic control and other health hazards. So most of our routine medications do help in the correction and prevention of muscle involvement in DM. Hence, my takeaway message is simple. Exercise, 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 and contribute as much as possible to a healthy diet. That will definitely take us up from diabetes, as also from sarcopenia, and probably from the cardiovascular assault. Thank you. Thank you for the opportunity once again.
Thank you, sir. Thank you for your wonderful speech. For want of time, we cannot do the question answer thing here. We can ask your questions during the tea time. And I thank all the three speakers whose speech was so wonderful and insightful. Thank you.
Thank you so much, chairpersons. For the next session, I would like to call upon Dr. Aravari and Dr. Gopinath to chair the session, handing over the session to chairpersons.